And thanks very much for the invitation. So I'm gonna stick with outsmarting vegetable pests, and we're gonna actually have a little more emphasis on the behavioral side. And that's going to involve one of the things you see represented up in the upper right-hand corner, which is a pheromone. And these insects scattered around are some of our subjects, but I'm going to let you stare at this for just a moment and see if you recognize any one of these. And there's one insect that doesn't belong, and the rest are subjects of my kind of tour of vegetable pests and their natural enemies that we've worked on. So, are you ready? All right. So, the banner up there is covering up the spine soldier bug. That one is a predator. It's preying on I believe, a cabbage looper there. And you see the one with the thumbs down, that's actually the minstrel bug, which is native to Europe. It's a umbellifer or Apiaceae or carrot. But it's oof, that coloration just lovely. We got a couple other kind of the usual suspects of some of the vegetable pests around here, and we'll be visiting each one of these in turn, some of them teamed up with their pests and natural enemies. And the reason it says squash bugs is one species, and we'll let you know about that too. So one thing, I'm going to get into some behavioral control, and what we mean that is using semiochemicals or chemicals that are used for in this case insect communication, for the possibility to modify the target insect's behavior. Now that could be a predator or it could be a pest. Most of the examples here I'm going to talk about are pests. Pest pheromone. Now pheromone is
Words there.
From another.
Everybody, mute your microphone, please. Sorry about that.
no problem. So, oops. I wanted to show everybody concentrates on, okay, we've got this, we've discovered this chemical that does a magic thing with insect behavior. Well, there's several stages in that, but there's many stages past that until you can actually apply it to any sort of pest management. So you see right up at the top, behavioral discovery. With the squash bug, we're at that stage because we know a male squash bug attracts both male and female same species squash bug. But that's all we know. We know there's a male produced aggregation pheromone. That's just the behavioral discovery or observation. We don't know what chemicals involved. That's number 2. Number 3 is identifying that. And number 4 is synthesizing it and confirming what you synthesized or made synthetically is the same as a natural chemical. And that all takes a lot of work. So if you have enough of that, maybe you can do a lab bioassay. You got to make more to be able to go into the field and test, hey, does this really work in the ecosystem that the insect exists? In other words, is there any meaning to the insect when you put it out in the field? And then I highlight this 7 because we've got a number that are ready for field deployment. That means we're ready to look at, can it affect, like getting down into 8, the populations that are out there? So a variety of ways you could use this. The way that most pheromones have been used for pest management in the past is the sex pheromone of the Lepidoptera or moths that of course are caterpillars as their immature stage. And you're disrupting the mating because it's put out by the female. And if you put a fog of it out there, the male can't find the female. Now, the ones I'm going to talk about here are different. They're male-produced aggregation pheromones. That means they attract both the males and the females. And guess what? That's really useful, and it's very different than mating disruption. So we'll get into some of those details. But as you can see, there's still several stages that some of the ones I'm going to report on are earlier than something where you would say, okay, Mr. Mrs. Grower, Ms., and put it out there. You know, does the grower find this useful? That's a huge step before commercialization. So as I mentioned, male-produced aggregation pheromone. These are common in the chrysomelid beetles or leaf beetles. And the ones you see here, if you can see my arrow, that's the Colorado Potato Beetle, the striped one. The other striped one's a striped cucumber beetle. And you can see they're right next to, you know, the brown marmorated stink bug. I didn't list the Harlequin bug. That was an oversight. But remember, there's always a wrinkle. So you think you have something very simple and straightforward? Guess what? You got to keep looking out for wrinkles because sometimes you might have something unexpected. Isn't that why we do science? We do it because we don't know the result. We want to find out something new. And sometimes it's surprising. Okay, just a little background on how you might use in management. Obviously, you can have a trap and monitor. You can say, I just want to know how many there are out there and have a good trap. The next step might be put a whole bunch of traps and try to depress the population. And another way might be you put out a crop and you put out this pheromone and you kind of supercharge and have a trap crop. Now you still, once you get the bug or beetle onto your crop, you still have to devise a way for getting rid of it, or it might just leave and go to your main crop that you want to protect. And there's other methods like attract and kill, including what used to just be called baits. Or you could lure the insect into an auto dissemination trap, which puts something on them like an entomopathogenic fungus that's pathogenic to them. And that's another way you might use it. I failed to mention there is Harlequin Bug there. I forgot. It was tucked away in the text. Okay, the first group of pheromones I'm going to talk about that was really exciting. And you know, I've worked a lot in insect ecology, but I'll tell you, the chemical ecology and discovering a pheromone, I tell you, that was one of the best eureka moments we had. You know, it just, when you get hundreds of bugs going to one trap and only a couple to the other, you know you're onto something. And this was work with Ashok Krumian, who recently retired as a research chemist, and the folks up at Carneysville, Tracy Lesky and her lab in West Virginia. But anyway, this discovery grew into the fact that Harlequin bug pheromone was discovered in the West by Jocelyn Miller's lab. Stink bug that had just invaded the East. So we wanted to look at this, but at the time we didn't know what it was. My internet connection is unstable, it says, but I think I'm back to normal. At the time in Japan, they were using this very different molecule from a different insect called the brown-winged green stink bug, Plaudia stali. And it was attractive to brown marmorated stink bug as well at certain times of the year, only late in the year. Well, that's not very useful. How are you going to tell if it's coming onto your apples or divert it from your apple crop if you don't know until, say, July or August or September? Forget about it. So, but that was the only thing we had at the time. And then once we discovered that the brown marmorated stink bug pheromone was a combination of these two isomers I'm showing here, Then I got the idea, well, we don't want to be telling growers to switch back and forth. We don't even know when these things are most attractive. So I thought, well, let's try the combination of the two and see if we might even get a additive or a synergistic effect. So let me just quickly review this chemistry. You see these things that look like little, I don't know, bowling pins or something. The tapered ones that are here, that indicates that in that molecule is a chiral center. That means a place where the molecule can either be one mirror image or the other. And if there are four of them, like there are with the brown marmorated
Are you big boxy?
stink bug pheromone, mergantiol, then if you think about it, there's 16 different combinations. And you see these little letters here, SSRS, that's the main stereoisomer because it goes SSRS. It's either R or S at each one. And the secondary one is RSRS. Anyway, all you have to know is it's complicated. Dr. Krimian worked it out, and I'll show you some of the implications. We got a twofer on this. Because insects evolve and they retain their own semiochemicals and pheromones, they tend to have related chemistry. And we got lucky that there's an overlap between the Harlequin bug and the brown marmorated stink bug pheromones. So as I say, we ran the pheromone with the methodecatrienoate, the pheromone of the other bug. And lo and behold, you can see from these green bars that every month it greatly exceeded the same dose of either one alone. So you can see MDT, the old pheromone that the only one we had at first, yellow one, and the true pheromone, they don't measure up to the ones that we put together. And we don't know exactly why this is. It's a weakness of my field that we can't interview these insects. We can't ask them, hey, why'd that smell so good? We have to interpret their behavior. And it looks like that's the best season long. So we had a season long, the combined lure. And this is now the global standard for brown marmorated stink bug, wherever it shows up. And believe me, it showed up in a lot of places. It's in Chile now, it's in South America. It's moved from East Asia to Europe. And it's, spread far and wide aside from North America. So I want to pause right now and ask for a few questions because at each section, I think I'll ask. Otherwise, you're just going to forget it and say, darn, I wish I asked that. Any questions at this point? All right.
I got one for you. So the mergantiol, is that effective at all on the squash bugs?
No, we're going to get to the squash bugs, but we don't catch squash bugs with it.
Okay.
Pheromones tend to be fairly specific. And one reason is, although I call it aggregation pheromone, the male is calling in the female. And it's to both of their advantage that they connect not with bugs of another species, but with their own species.
Of course, yeah.
Sometimes we see interaction, but they're usually pretty specific. And as I mentioned, we had a wrinkle. The attraction is synergized by this other pheromone of this other bug species, which does, when I say sympatric, it does coexist with brown marmorated stink bug in Japan, China, and Korea. So they've evolved together. We think that the brown marmorated stink bug is using the pheromone of the other bug to help find food that they both find attractive, and in the fall to find good overwintering places. And if you know your house, rocks, hollow trees, these are all brown marmorated stink bug overwintering areas, cavities, whatever. Okay, Harlequin bug. So the commercialized pheromone is actually a mix of all those ones I mentioned, 16, and lo and behold, both the Harlequin bug and the brown marmorated stink bug are attracted to it. They're only slightly sensitive to the blend, so we don't worry about that. But with the Harlequin bug, all right, time to mute. I don't know why they keep coming on. So with the Harlequin bug, the wrinkle is to get the maximum attraction and to use what is frankly cheaper material, we should use the mustard oil because they are whole crop specimens. Here's the wrinkle. So we're going to make the brown marmorated go away and focus on the Harlequin bug. And the Harlequin bug is colored aposematically. That means a warning coloration. Whenever you see orange and black like you do with a monarch and a lot of lady beetles, and there it is, again, with the Harlequin bug, you're gonna expect it to be very conspicuous, and it's basically warning predators, I don't taste good, better not eat me. And the reason they don't taste good is they retain the mustard oil. And those are called isothiocyanates. And I had to practice that for a long time, but now I can say it very easily. Anyway, the wrinkle is that if this plant chemical is present, the pheromone required is very low. You just need a little bit.
Excuse me for 1 second. Everybody, mute your.
You can mute them, I think, Fred. If you're the boss, you can.
Well, I'm doing it, but I can't find them in time. There's like 50 people on board here.
It's quiet now.
This was not one of the students, though, I should add.
That's no problem.
Sorry.
So with the isothiocyanates, because these bugs are specialists on mustard family, they home in on that. And what is the best idea of why that's effective is the male is calling or emitting the pheromone from a plant that smells like the mustard. So that's all the female needs to know is I've got an honest male calling me from a good host where I can mate and lay my eggs. So we decided to use this a little bit, maybe theoretically try and figure out what's stronger, which signal is stronger. So when we added pheromone, we were trying to kind of fool the Harlequin bug. So if we had a collard versus a collard with a pheromone, well, the pheromone went out enormously. See how much more it is there. If we had a soybean and a collard, a soybean, they're not interested in soybeans, so they all go to the collard. Now, if we put it, both have a pheromone, they go to both. If we go with the preferred host collard and add a pheromone, it's even stronger, right? What if we do the opposite and add the pheromone to the soybean? Well, the pheromone's a pretty strong signal, so we can actually fool it to go on the soybean. Now, these were just sampled twice a day, so we know that bugs come and go. So when you see a pattern that has 30 per plant on the soybean, like you see the bottom bar, and only 5 on the collard, that's not necessarily how many arrive, because we know that what the numbers we see relate to attraction and retention. So some are attracted and then they taste the soybean and literally leave within minutes. So to address that issue, we compared it. Well, this was in a publication. I like nice titles and it was appropriate. So it's Fooling the Harlequin Bug. And a harlequin was kind of like a jester in the old days. That was a synonym. So we thought that was a fun thing to say. And so we took and put a systemic insecticide in the plants so that when the Harlequin bugs came in our test, they tasted it and they dropped dead. And if you can see, when we did that, just like our first trial that I showed on the previous slide, we were able to fool the Harlequin bug to go to soybean. But if we poisoned it, we got 269 times the response that we did on the collard versus 3.8. So you can see a lot of them are arriving and then leaving. And what does that say? It says if you're going to use a trap crop that's not a very good host, you better knock them dead right then, because you're going to have a lot departing. Now you can see down here, we didn't get any indication of preference when we added a different isothiocyanate. The response was not significant. That's what NS means. And lo and behold, when we did the same thing and added a systemic insecticide, we got roughly 25x had actually been on the soybean and then left. So this is an illustration of how complicated and multi-stage an insect behavior can be. So questions on the Harlequin bug? All right, I'm going to briefly visit the Colorado Potato Beetle. It's an infamous insect pest, and I'm going to say why is because it's evolved resistance to probably more pesticides than any other single insect, at least in agriculture. Probably the mosquitoes have it beat, but agricultural pests that are notorious for pesticide resistance, this is at the top of your list. And several years ago, now actually 20 years ago almost, Dick Dickens of our lab, who's now retired, he discovered that this molecule here was the male-produced aggregation pheromone of the Colorado potato beetle. And I got a little S there that says what the isomer is, because the other one's not active at all. The R isomer isn't. And he called it, and I hate this name, SCPB1. Now, how's anybody going to remember that? So I'm going to call it doryphoridiol. And I'm not going to explain why, but after you say that a few times, you'll understand you'll get that, you'll get it. Anyway, this molecule is attractive to both sexes, and we had several grams just synthesized, and we're going to do some trials this coming season. So it's possible we might use this as a trap to supercharge a trap crop or an attract and kill. It may be useful to have the green leafy volatiles, but it doesn't seem as important as with the Harlequin bug. But what we discovered, and it's in the literature, and I knew this with some Hungarian folks that I worked with when I was over at ETH in Zurich, Switzerland. There is a female-produced sex pheromone. You can tell because the males will come. And here's how we found out. So remember I told you about, we don't use a lot of systemic insecticides, that is neonics. We don't use them out in the fields, but we do use them experimentally. And if you'll see this setup, we have beetles inside a screened plant, and this is just a paint strainer you go to Home Depot and get. And it's very fine screen, and it keeps the beetles in. And then next to it is a companion plant that's a potted potato plant that's treated with imidacloprid. It's poison. What happens is the beetles come and they can't get into the one that's screened, and they go to the one that's not, and you can see them piling up here. Okay, so when we did the male beetles, the males were enclosed in the screen here. You can see we attracted, compared to none over here on the right, we attracted males and females. When we use our colony males, they weren't quite as vigorous. but the ones that are the bright colors, it's attracting both males and females and more than the blank. And the reason it isn't a whole lot more is Colorado Potato Beetle is pretty good at sniffing out potatoes. So the pheromone relative to the plant signal is not quite as strong as what we saw with the Harlequin bug. Anyway, if we put out females, we only get males. So that's known as a sex pheromone. And we believe this is the first beetle which has been shown to have both a male and a female produced pheromone. Beetles are they're the most abundant order of insects. So that was kind of a cool thing. A lot of others, like the boll weevil, are known from male-produced aggregation pheromone, or their male-produced sex pheromone, or their female-produced sex pheromone. But we never saw a species yet until this work that my postdoc Ariella Haber did with both sexes two-way. We think it's more common. We think people don't realize it and have overlooked it in the past. So, and we saw a dose response amongst the males to the number of females there. The dose response had already been shown for the aggregation pheromone. But when we looked at how many females came into the females screen plants, they didn't care. So it's a sex pheromone. It's only attractive to the one. All right, that's Colorado Potato Beetle. Now I'm going on to my big topic. And so I'm going to ask for questions on anything up to this point, and then we're going to start talking about cucumber beetles and other cucumber pests. All right. Speak now or forever hold your peace. Striped and spotted cucumber beetles. These are the most damaging pests of cucumbers, especially in the east. And the reason they're damaging is they're very good at finding the crops, and they cause heavy damage early on when the crops are vulnerable. If you direct seed, sow the cucurbits, and I mean squash, pumpkin, cucumbers, even watermelons, melons, etc. If you sow these directly, these striped cucumber beetles especially will cause heavy damage to seedlings. They are very much a specialist. They are on the cucurbits, that family of plants, which are largely native to North America. And we'll get back to that. Here's some spotted cucumber beetles, and they do like cucumbers and other cucurbits, but they have a wide host range, 200 or more flowers. They're all over a lot of things, but they like cucurbits because they have a bitter compound called cucurbitacin in them. We'll get back to that. They're both all the way, they either have a sister species or subspecies out west. Importantly, for growers, including gardeners, these species both transmit a bacterial wilt that's very damaging, especially to cucumbers, but to some other cucurbits. And here's Exhibit A. This is the wall of my house in Arlington, Virginia. And it has happened more than once, but this was the best picture. So here's my beautiful crop of cucumbers just starting to flower. Unfortunately, you can see a few little wilted leaves there. I have been diligent and tried to remove those cucumber beetles, striped cucumber beetles, but I wasn't diligent enough. And you can see this is only just more than 2 weeks later. Look, that's the same thing, and they're not going to yield a single cucumber. So a lot of people have given up growing cucumbers because look what happens. You're disappointed, right? So we're looking at a way that we can depress the populations locally. We're not looking to wipe them out or eradicate them. That's unrealistic. But using the pheromone, we might be able to make some headway. So what are we talking about? some years ago, it's now 20 years ago, that my good friend Rebecca Smith and Mike Hoffman up at Cornell University described, discovered, and synthesized this little thing that looks a little bit like a dairy cow. It's beta-lactone. And the structure is involving these cone-shaped indication and this little dotted line. They have 5 stereoisomers, so it's a complex molecule. And the synthesis is difficult if you want exactly that one. But there was a discovery that if you just keep this, the chirality or the mirror images right within this ring, then they don't care if it's a mix. They'll only respond to their pheromone, but they don't care about the other ones, which is very different. If some of you know about Lepidoptera pheromones, many of them are very sensitive to the ratios and the isomers. Not so much with these. We're lucky because it's a lot cheaper to make a mix like that. So what's happening here is the males go out. In the case of the striped cucumber beetle, they find a host and they produce beta-lactone, and both the males and the females are attracted. Now, why hasn't somebody asked me so far why would the males want to attract males? Well, it's not just the sender that decides that, it's the receiver. So if you think about the incentives of these beetles, a female wants to come in.
Okay.
Desmond, would you mute your thing there? Okay. So a male emits this pheromone, the female wants to come in and mate and lay her eggs because they specialize on these, feed and lay her eggs. The larvae are grow up on the roots of cucurbits. Okay, the males want to come in because heck, the females are there, so they're going to come in anyway. And as my soon to join fellow scientist Anna Wallingford would say, they want to join the party. Okay, so I mentioned earlier this very large molecule over here. It's one of the cucurbitaceans. If you've ever tasted a bitter melon or some melon blossom that's bitter, these chemicals are common in the wild cucurbits. They're not common in the ones that we eat because most people don't like bitter. They've been bred out of cucumbers and other things that we eat. So, but the beetles love them, and the spotted cucumber beetle does too. So if we can combine the pheromone as a distance attractant and the cucurbitacin as a feeding stimulant, gosh, we might have a good attract and kill tactic, or maybe just might call it a bait. Okay, so we're right in that 7 to 8 area with the striped cucumber beetle. So we put this out and we did the simplest, dumbest thing you can imagine, which is hang the pheromone lure next to a clear, sticky trap. It's messy, but it works. It's quick and dirty. And look what it did. 1 milligram of the mixed beta-lactone versus a blank. That's your control. And look at that. It attracts about 8 times as many. And this was true across the whole season from May to December, or almost December. Anyway, but we got a great surprise. The spotted cucumber beetle, which does not produce beta-lactone, and 2 species of squash bug, the squash bug and the horned squash bug, were also attracted. Well, wow, why is that? We'll get back to that. But they're eavesdropping. It's not their pheromone, but they are smelling that. And remember, this is a native crop, and all of these herbivores, which we call pests because we want to eat it, not them. All of these pests, all of these herbivores are native to North America. And you know that the Native Americans have been raising cucurbits for a very long time. And even, well, you get a dose response. You expect that with the striped cucumber beetle, that's its pheromone. But we get a dose response from both sexes of the spotted cucumber beetle and both sexes of the horn squash bug, and at least the females of the regular squash bug, the squash bug, which is another species. And you can see there's a dose response here. Okay, that was exciting. And maybe we shouldn't have been so surprised, but man, we were excited. Okay, so here's what we had. We had the pheromone beta-lactone, we put it out. It's produced by the males, and the little kind of horseshoe-shaped arrow indicates it's attracting both sexes. So that part. And then I just told you there's these other 3 species that are attracted as well. And the reason we think they're eavesdropping is the striped cucumber beetle is notoriously effective at finding the food that they all want, the squash plant. But we don't know the whole picture yet, and we're working on some more. So we were working with colleagues from UC Davis, and the western striped cucumber beetle produces beta-lactone, and of course, the males and females are attracted to it as well. But that wasn't the whole of it. We think there's more to the story, that natural enemies like the tachinid parasitoid Trichopoda penipes might be attracted, and the parasitoids of the striped cucumber beetle, Celatoria, might be attracted, and we got to see. We also know, as I mentioned earlier, that the squash bugs probably have their own male-produced aggregation pheromone. And we know that the spotted cucumber beetle has a female-produced sex pheromone. So there's all sorts of stuff going on before we even add, and I've got it just as a cloud, floral and foliar volatiles, which are produced by the plant. And those tend to be simple compounds that are cheap to produce, whereas beta-lactone is not cheap to produce, at least so far, even with the fact that we made it a little bit of a messy mix. All right, so why do we call this a keystone semiochemical? I just made that up. If you find something really exciting, you get to make up a term, right? So as I mentioned, New World squash and pumpkins are native. They're native, and they're a native agroecosystem. And they've been grown by the Native Americans in Mesoamerica and up into North America for a very long time. And those are part of they support these herbivores. And why should we be quite so surprised when they're eavesdropping on one another? So when we do have to make this distinction that the cucumber and the muskmelon and the watermelon are old world species, so they may not work the same way. But can we leverage what I call coevolutionary based semiochemistry to maybe come up with some good selective pest management? So we did some of the standard things and said, well, let's find out about what trap might work real well. We started with a clear sticky trap, but if you've dealt a lot with sticky traps, you get sick of them. They stick to you and you have to wash it off, and the insects are a mess. There's not good specimens when you want to find out what they are. You want to find out if they're male or female. It's a mess. So we used several things, including the boll weevil trap, this green bucket trap, which is a complete dud. And we compared the yellow sticky trap. Well, the yellow sticky trap is attracted to a lot of things like lady beetles, grasshoppers, bees. We don't want that kind of thing that's so non-selective. We want less non-targets. Even if we didn't care about the non-targets, the sticky panel is going to fill up with insects, and it's not going to be able to stick to the one we want, which is the cucumber beetles. So we found that the ground-based boll weevil trap was really good. And the openings are too small to let in bees, and it's apparently too low to attract a lot of other insects. But if you think of the squash plant, especially a squash seedling, here's the striped cucumber beetle coming in. It's like right there and it's perfect. And for whatever reason, they come in real low and then they want to go up. So we don't know why, but certain insects like to go in a bucket trap and go in and go down. Others probably has to do with the way they encounter their host, like to go up, and they like to go up. And remember that bucket trap was a dud because they don't like to go down. Okay, so this is the non-sticky contender for replacement of our sticky trap. And if you can see here, it's a complex graph, but the clear sticky trap and the boll weevil trap, the statistics say those means are not significantly different. Over here with the spotted cucumber beetle, a little different. They love yellow. If you grow yellow flowers like yellow zinnias or yellow mums, they're obnoxious. They get inside the blossoms, and the poor you know, roadside stands that are trying to sell or have you pick blossoms have these spotted cucumber beetles in it. And you can see the white sticky, the yellow sticky trap that wasn't even baited caught more than the baited clear sticky trap. Oh my gosh. Anyway, nothing's perfect, and we had to settle on the boll weevil trap mounted on the ground. And we did a little work with it. Where are these beetles? Well, at the beginning of the year, look at the left graph, and this is before the squash is even out there, there's large numbers of striped cucumber beetles, and the dotted line is the female percentage, which is at that point it's about a third of them are females and about 2/3 are males. Remember how I said the males are the ones out there trying to find things, and they're going to call, and eventually the females come in. And then there are 2 generations. So the second peak in September, October, that's the second peak. And then we did this more detailed examination later, and we find, look at all those ones we can catch in the border before either corn or the squash is planted. So that's our key time window where if we wanted to produce a local depression of our pest population, that's when we do it. And guess what? Once they get onto the cucurbits and they're in the squash, they found their plants. The other beetles are there. They probably have mates. They have a place to lay. They don't want to go in a trap. Why should they respond to V-titolactone? They've already found their host and probably their conspecific beetles, male and female. Okay, so we can see that pattern. And with the spotted cucumber beetle, it's a little bit different. But we've only got a limited time here, and I'm going to tell a little more of the story. So this is, we always think of cucurbit flowers, and they're beautiful. This is a cucurbit flower, but it's not bees inside. Guess what's inside? The striped cucumber beetle is eating the pollen and eating the nectar. And guess what? They are actually lowering the pollination. Even if a bumblebee comes and very efficiently pollinates that, or a squash bee, it's not as likely to set a good size pumpkin or squash fruit because the beetles are in there mucking it up. And believe me, the bees don't even want to go in there when there's that many beetles. Here's one of the ways where we thought about using this to advantage to reduce the number of beetles. And we're working on some other things too. We called it the roller derby. And one of my colleagues up in Vermont said, hey, how are we going to present this bait? And we want to know how many beetles come. And we're going to have a bait station. So he said, well, why not just soak a paint roller? And so that's a paint roller. That's a pie tin. That's a roof shingle. This is a binder clip. There's not a lot of expensive stuff here, except that lure costs about $4 a piece. They last for 2 weeks. We wanted to see what's the importance to these 2 cucumber beetle species of V-titolactone, just a plain melon juice, and this is watermelon juice, sweet watermelon juice, And then we have a special bitter watermelon juice that contains this cucurbitacin. But our colleagues out in South Dakota made it pure for us so we can test each one independently. And this is a three-way factorial experiment. So there's rollers with nothing, there's rollers with plus or minus beta-lactone, plus or minus melon juice, or plus or minus cucurbitacin. So each factor is tested. And lucky us, there was no what we call statistical interaction. So they all just responded in a simple way and not interacting. And surprise, surprise, the striped cucumber beetle responds 14-fold to beta-lactone compared to those rollers without it. And these are replicated and blocked. You can see them along the side of the field there. And they also respond 4x to the melon fragrance and the cucurbitacin. Once they land, they start eating it. All of these were laced with an organic approved pesticide, which is known as spinosad, brand name and trust. So once they start eating this, they're gonna end up in a little pile on my shingle. And the spotted cucumber beetle loves that cucurbitacin. And the reason it does, we think, is many of them, the larvae don't accumulate it. And they need cucurbitacin. The males in particular give it as what's called a nuptial or mating gift to the female to help protect her eggs. Remember, that's bitter cucurbitacin. But as we found with the traps, they're very attracted to the beta-lactone and also significantly to the melon juice. So this would be expensive and kind of a hassle for growers. So we're working on some different ways that this could be more grower friendly. This was more what we call a pilot study or just a proof of concept that these are all important components. I think I'm gonna pause and ask if there's questions about the cucumber beetle work. And how are we doing for time, Fred? Because it's getting.
You're fine. You're fine on time. No worries.
Okay.
These pheromones, you mentioned, they're pretty expensive to produce, right?
Not commercially available.
Not even, okay. And like these rollers, how big of an area would one of these rollers treat? So just to get an idea of the cost here.
Don't have a good, we're working on that with Virginia Tech and Tom Kuhar and his graduate student Demian Nunez to look at what the plume width is. We think the beta-lactone is drawing from probably 30 meters away. And that's why we space these out. But we're not sure of that. And it's kind of hard to test. Well, our bottom line though is, can we encircle a field even before the crop is planted and depress the populations compared to another field where we didn't do that? Well, that takes a lot of work because you've got, a full field is your experimental unit. You want to replicate that. You probably want to replicate at least 4 times. Right there already, you're up to 8 fields with your control and your best guess at your best deployment of this. So
how big is a field then?
Well, we're using with Demian's work a 1/4 acre. In some cases, it's going to be literally a community garden where we did it or we didn't do it. Remember, I said that gardeners are having a heck of a time, especially with their cucumbers, with the bacterial wilt that they vector. And we're seeing if we can essentially, we're not going to trap them out completely, but if we reduce the numbers enough, we may be able to be successphylla.
What the bacteria, what is the name of the bacteria involved with the wilt?
Erwinia tracheophylla.
Erwinia,
That is referred to, if you look in the literature, as bacterial wilt of cucurbits. And it's a really common pathogen in the East. And
I've seen the damage all the time.
Gums up essentially gums up the plant's water vessels in it, and that's why they will. It's really a sad thing when you see it happen, especially to your own crop.
Is it only cucumbers or other cucurbits as well?
It's variable. We have a profile in Journal of IPM, which is an open access journal of Integrated Pest Management, which if you look at the striped cucumber beetle profile, it lists which ones are susceptible. Cucumbers are almost all susceptible. There are a scattering of others throughout the cucurbitaceae. Some are resistant and some are susceptible. But I don't know about you, but I don't want to give up on cucumbers. I'd like to be able to grow those.
Sure.
Okay. So, bottom line, we think that the distance attraction of butyrolactone, the feeding stimulus of cucurbitacin might be a good thing for the bait. And now we're trying to stretch this expensive pheromones with some very simple floral or foliar volatiles that are produced, of course, by the plants. The only thing is, watch out, because other things smell those, like bees, and especially with the floral volatiles, so you have to be careful. So we're testing that. I'm going to switch gears a little bit and look at a different squash pest, which I just mentioned, the squash bug, Anassa tristis. And here are here's an egg mass. And here's an adult squash bug on black plastic, which a lot of growers use to heat up the soil and grow good squash. The problem is the squash bug love to hide in the holes in the black plastic, and they do quite well. And when they do quite well, their eggs hatch, and you can see all these little nymphs. And these bugs can make a mess of zucchini and some other cucurbit crops, particularly in the Peppo group, which is summer squash and zucchini, and also includes many of our pumpkins. Now, when that happens, that fruit may not be marketable. And also these vector another, they've been found recently to vector another disease. I'm trying to remember what it's called. Oh, geez. I'll get back to you on that. It's a bacterial. So when you see these leaves like this and hear the nymphs, you got some trouble, and they love to hide in these holes in the black plastic too. Here's the hero of the story. A very small, remember that egg? That was very small, just about 1 millimeter in diameter. And this was, remember, that's what the egg mass looked like. And here's the little guy, used to be called Gryon, now called Hadronotus pennsylvanicus. And some of the research that I'm going to show is mainly that of my postdoc, Sean Boyle. And this photo is from Elijah Talamus, who used to be down at the Smithsonian as a postdoc with Matt Buffington of the Systematic Ent Lab, is now with the state of Florida. Here's Hadronotus pennsylvanicus, egg parasitoid. For those of you that might not know what an egg parasitoid is, they lay their eggs in the squash bug egg, their host. You can see, here's one doing it right now in that picture. And as a result, out comes a little mini wasp, not the squash bug. Great for biocontrol, because the one that emerges, and most of them are females, they go out, find more eggs, lay more eggs, parasitize more eggs. Here's a set of the horn squash bug. They have a little different egg pattern, and they look kind of bronzy. And look at those. Every single one has a hole in it. That's where the wasp came out. And no horn squash bugs came out. So that's a very effective egg parasitoid, and it's been found at pretty high levels, but not early in the season. We don't know why, but they don't seem to overwinter very well in many areas. They overwinter, but not enough to get started. The squash bug gets started, damages the summer squash and zucchini and pumpkins. Growers end up making chemical treatments, and this guy has noticed. So here it is again. It parasitizes, as I showed, most of the egg mass, high fecundity. If they don't lay eggs, they can live for quite a while. If they lay eggs, they don't live that long. They do overwinter as adults, though, so they live quite a while over the winter. And there are only a few males amongst them. This is very common with the parasitoid. Sean has worked out how to rear them in the laboratory. And as I mentioned, the natural parasitism is very low early in the season. So if we want to give this a head start, we're going to do something called augmentation biological control. Augment just means we add to it. And when we do it is early in the season. And here's Sean with some of his growers on the eastern shore of Virginia, where he did his thesis work. And here's the result of the release, which was in the squash fields in these organic farms on the eastern shore. And you can see the no release, they're really lagging. There's not much parasitism. It's in the 10 to 25% range. Whereas the release provoked a significant increase up to about 75% of the eggs were parasitized. And remember, those nymphs, then they're going to be subjected to predation, and also the next round of eggs are going to be parasitized by the same egg mass. Here's a group of them, and these are actually multiple females there. And some of the things that Sean's been trying to work out are how many do we need, how to distribute them, can it be feasible, economically feasible? And I don't know if some of you probably know the New Jersey Department of Agriculture up in Trenton, they actually rear parasitoids. They rear a parasitoid for the Mexican bean beetle. It's a multi-year program that's been super effective, and they've reduced that insect, which is non-native and a big pest of soybeans and common beans. It has almost vanished from New Jersey because they've been releasing the parasite, and I can't remember the name of it. It'll come to me. Anyway, they're interested to rear this because the New Jersey growers grow a lot of squash, Garden State, right? Okay, the other questions are, what guides this parasitoid, and can we help it find the eggs? It's pretty good, but on the other hand, like I said with the pests, sometimes things will be attracted and then they'll leave. And one of the reasons they might leave is they have another host called the leaf-footed bug or Leptoglossus. And across the top is the Anassa or squash bug, and here's the Hadronotus again. But in the bottom is the leaf-footed bug, and their egg mass looks very different. You can see them all lined up there like little barrels. And guess what? That Hadronotus really likes the leaf-footed bugs. And they're much more of a minor pest in the East. They're looking into it with the pistachio and almonds that the other leaf-footed bug species are pests of in California. So we're working with that group, both on the Hadronotus, and there's now an aggregation pheromone known for leaf-footed bug Leptoglossus. These were some of the lab outcome of choice tests between the Leptoglossus, which is the leaf-footed bug, and the squash bugs. And lo and behold, they preferred the leaf-footed bug, even though they do fine on the Anassa or squash bugs. But we have to know this. We have to know the ecology and what their alternatives are out there, because we might release some, and if they don't find the squash bugs, they're going to go away and look for leaf-footed bugs. All right, there are some other, this is Hadronotus pennsylvanicus at the top. I don't want to go into all the details, but they are a parasitoid of multiple Leptoglossus and Anassa species. And we don't know whether maybe they're different populations because they're all over the place in terms of geography. There are tachinids, including this very, very beautiful Trichopoda penipes that have a broader host range. They go to penatomids and they respond to those pheromones. So remember, I had one of those screened in pots with we had a squash plants screened in like that with the squash bugs on it. And I threw some old ones in the pickup truck, brought them up to my lab, which is half a mile away. Guess what's there? The tachinid. It's smelling. They have for sure a male produced aggregation pheromone, but it was fascinating to see that. And this molecule represents the leaf-footed bug pheromone. The squash bug pheromones are not known. So here was some work that Sean did to try and figure out, was the Hadronotus pennsylvanicus, which is abbreviated here HP, attracted to the area when we put the Anassa species, the Leptoglossus, and just for fun, we did a V-tadalactone lure to see if they were eavesdropping on that too. Well, we didn't get a lot of effect with the parasitoid itself, but we proved that, lo and behold, they're coming into their own aggregation pheromone, and they're on the outside of the screens, which you can see there. So that's to be continued. So I want to wind up and just emphasize that all this is a lot more complicated in my book, much more fascinating than treating with pesticides, which is the predominant way that squash pests are managed and most vegetable pests. But if we can integrate the semiochemicals, including the pheromones, with biocontrol, we need this careful evaluation, and we need to make it grower friendly so that at least some growers are willing to try it out. And if those growers are successful, we can hope to spread the word. It's very important to know that ecology, to know what things are affecting the insect behavior of both the pests and the natural enemies. So to do that, we need to know all that to make them compatible with what's happening with growing healthy crops. I want to thank a lot of cooperators that we've had through a variety of universities and grants and the assistance over the several seasons of work I've showed you in our farm crew at Beltsville. And I'd be happy to answer some questions.
Well, thanks, Dr. Weber. This is really informative and very eye-opening for people who are unaware of all the different things that are out there. When you deploy the pheromones, how long do they last out there in the field?
As a rule of thumb, when we put out the first what's are called rubber septa, they're just a week. We feel like typically we can get. Then after further testing, maybe we get 2 weeks. Then if we want more, we work with the pheromone companies, and they're small companies, and we know who to work with. They often have a very good time release formulation that they could mix up. One company we're working with now is called Iscatec. They're based in Riverside, California, but they have operations in Brazil as well. They have a formulation they call, I like good names, Splat. They call it Splat. It looks like it has a viscosity all the way from like a latex caulk, like you caulk your windows with, all the way out down to, say, latex paint, which is quite liquid. And this holds a pheromone, typically shields it from UV radiation and rain, and can function also as an edible bait. And so it's all in one. They formulated, one of their first formulations was with the moth that used to be called gypsy moth. It's now called spongy moth. And they wanted to do a mating disruption. So the researchers were thrilled when Iskatech formulated it as literally a paintball. They had paintballs filled with a splat, and the researchers wanted to shoot them up into the tree canopy because that's where the moths are. But they couldn't get that splat with a simple, say, caulking gun up there. So they were able to shoot it up with a paintball.
That's
gun.
great.
And they had great fun, and they also had good mating disruption. So we're working with that company to see if we can integrate, in a time release formulation. We're not the formulation time release experts, but we get enough good results like those that I showed you with the comparison of the traps. Those were replaced every 2 weeks because some data that we showed did earlier where we aged the lures. Fred, we then compared the aged lures to the new lures, and we knew we had 3 weeks. And we said, well, let's play it safe. This is, research. We really want to know the results. We'll just do it two weeks. And it was fine. Good question. Do we have anything in the chat? Mostly it's just people identifying themselves. Okay.
Yeah, and some thank yous and that sort
How
of
did
thing.
Native Americans control these cucurbit pests? Ha ha! That is a great question. I don't know how to answer that. I'm not even sure we can. We do know that they were oftentimes grown in shifting patches that were polycultures. And when we talk about polycultures, one of them was the three sisters: squash, beans, and corn or maize. And that in itself tends to discourage insect pests because they have a trouble finding things. And there's also a lot of refuges for predators that might eat them when they come. However, we also know that some of our cucurbits, like Cucurbita maxima, that's a species that includes some of the big pumpkins and funny looking Hubbard squash, and some of the other cucurbita, they're more resistant. And some of our modern cultivars, and Yolanda Chen up at University of Vermont, David Hawthorne at University of Maryland, been working. What do we do many times without realizing it? When we domesticate crops, sometimes we make them more vulnerable to the pests. So I can't answer the question. It's a great question, but I don't know if we're ever going to find that out. But I will say that the Native Americans grew the squash in a very different context. So polyculture and shifting culture as well. And if you think they didn't know about these pests, yeah, I'm sure you're mistaken. I'm sure they knew about it, and I bet you they did have some strategies. And maybe, frankly, they weren't as greedy as we are in terms of wanting every last bit of yield for every last thing. There, some of the tribes, at least, attitudes toward nature were more, okay, we're going to share some. There's not a need to harvest every single squash. Any more questions?
Anybody like to ask a few more things? There's a ton of questions that could be asked, I'm sure. I was impressed with the photography and those hydronotus hatching out of the eggs. Great picture.
So that's one of these. I'm gonna page up to where it is, because I agree with you. So
Yeah.
That was Elijah Talamus, and they have, I'm trying to remember the brand name of it, but it's very well known amongst the people who take close-up shots. There's a way that the microscope moves in and out of different planes of focus. And then there's a program that integrates all of those. So the depth of field is a combination of all those images.
Stacking.
It's a stacking algorithm. And so that's able to do that. You know, I find it super impressive myself. Yeah.
It is, yeah, it's very nice. Yeah.
And they have that with the systematic ant lab. And then we have a microscopy lab too on the campus at Bark, where they do that. They do a lot of other things. They do scanning EMs, which are fantastic. So my late colleague, Mary Cornelius, who also worked on this insect, she had them determine the eggshell thickness using the scanning EM down to the, you know, it's microns, very, very small dimensions. And she found out the Anassa tristis, which is the squash bug, has a much thicker eggshell than Anassa armigera, the so-called horn squash bug. And that accounts for the fact that this parasitoid takes a lot longer to drill into and process, the handling time for each egg of Anassa tristis, which may account in part for the reason that Anassa tristis, the squash bug, is definitely more of a pest. It may be that Hadronotus is not parasitizing it as effectively. So there's a lot of uses, and yeah, I agree with you on the photography. Some of these other ones were, oh gosh, I don't even know who took the striped cucumber beetle. I think it was I think that was one from Purdue, from Ian Kaplan at Purdue. Okay, any other questions?
Yeah, the chemistry work.
What's that?
Chemistry work, all these characterizations of these pheromones and all that, and knowing where the chiral centers are. Where does all these are probably stuff that's been in the literature for a while, I'm thinking. But when you guys work with them, I mean, who does the chemistry work? Is that your lab or another
Well,
lab?
it was Asha Krimian's lab. We went from 3 chemists down to 1. His name is Ajin Zhang, but he did not work on this project. We're hoping to hire another research chemist. And we've also worked, I think I mentioned at South Dakota State, we have a colleague who specializes in the larger molecule type pictured here, the cucurbitacins. So, you know, you work with the chemists that you know how to deal with it. We're working on the leaf-footed bug pheromones with Dr. Jocelyn Miller, who's at UC Riverside. There are actually very few pheromone chemists around, and it's really too bad because the leading countries in the field are the US, Germany, Japan, with a few others, particularly in China and Sweden. But there are not a lot of chemical ecologists, as we call them, that have the skills to know, you know, both the chemistry and some of the background with the organisms. One important thing in this regard that we don't have technology in our lab very well developed, but it's important, is to take the antenna of the insect and use it as a diffuser. And a subset of the chemicals that provoke a nerve response, a sensory response, a subset of those is the ones that they actually act on. So we had electro antenna detectors of that the Cornell people had done on the striped cucumber beetle. And then the Riverside folks tested it for the Harlequin bug. And then we still had, no, I'm trying to remember. I think it was the Riverside lab also that did the EADs for the Brown Marmorated. So anyway, we have to treat our chemists well, and we really appreciate all their work. And then there's the synthetic side, which is a whole other challenge.
So, Don, one of the students just asked about how often do these bugs reproduce?
So in our area, the chrysomelids and the stink bugs are both usually 2 to sometimes a partial 3rd generation. So, you know, in our perspective, maybe that's fast, but compared to aphids or whiteflies, which have maybe 7 to 10 generations a year, it's not super fast. So a lot of times a squash, like for instance, summer squash, if we could knock out the 1st generation, and I don't mean eradicate, but I mean knock it down to a very low level, we would be happy with that and not so concerned with the 2nd generation because they're not going to build up that quickly. So that's a very good question and a good perspective. Now, the Hadronotus pictured in the upper left here has multiple generations, and as a result, can cycle through 2 to 3 generations for each generation of squash bug. So we really like that with a biocontrol agent, in that it can respond and have what we call a numerical response, as well as a functional response. So that's a very good question. Thank you for asking it. I should have made that more clear in the start of it.
Ed, do you have a question for Dr. Webb? I see you have your microphone off. Did you want to ask something?
Oh, are you talking to me?
Yeah, you're Ed Cohen, right?
That's right. Okay. No,
How are you?
I'm just listening in. It's interested in what he has to say, and
Just wanted to make sure if you had a question or not, that's all.
no, I don't really. No, I don't.
Somebody said that the bugs are really pretty and you'd like to make jewelry. People already make jewelry out of bugs. In fact, you probably have heard of the emerald ash borer. It's in the flat-headed wood borers or Buprestidae. Their relative, larger relative in Thailand, they make jewelry out of the elytra or hard forewings, and those beetles are eaten as food there, and then the elytra or the wing covers are used to make jewelry, and no beetles were killed for that purpose because they're already eating them. But anyway, they're very pretty jewelry, and if you come to an entomology meeting, there'll probably be a vendor selling that. And there's a lot of other, you know, pretty insects, but I would say those are prettier than any of the ones I showed.
They eat the Buprestid beetles, really?
Yep.
I know, I've heard of that in the Kalahari Desert, some of the Buprestids
There are a
there.
number of what we call entomophagous cultures. We've gotten away from that a little bit. Maybe we'll get back to it.
I like shrimp.
There's a lot, yeah, you know, kind of like the shrimp of the land, if you expand your viewpoint here.
Yeah.
We do eat cicadas when they come out. We may have a minor emergence this year. We're hoping to see some in our area. And in that case, the newly emerged cicadas, the ones that are still white, you got to get out early in the morning. Those are the best. With tempura. And here's a warning though, if you're allergic to shellfish are arthropods, they're crustaceans, they're not insects, but they're closely enough related with the chitin, and some of the proteins overlap with insects. If you're allergic to shellfish, don't go chowing down on cicadas. Maybe take a nibble and see. but don't make a meal of it. You might get really sick. So
Is that
we-
right? They overlap. I had no
yeah,
idea.
the allergens do.
That's amazing.
Depends on your specific immune reaction. Just
Right.
like I'm pretty allergic to honeybees, but yellow jackets don't bother me much. It's partly a question of the volume of poison, but it's also what type of toxin it is. So the spotted lanternflies, somebody asked, are they expected to emerge again this year? I am not a spotted lanternfly expert, but I know that I was surprised for the last 2 years that we haven't gotten, because they've been very close. They've been in Winchester, they've been up north of Baltimore. We're getting them now. We're probably going to see a lot this coming year. And they're a completely different bug than what we've been talking about. But there are other people, particularly at Penn State, but also at Virginia Tech and elsewhere, working on them. And one of the commonalities is it looks like those eggs, which is the overwintering stage for the spotted lanternfly, are vulnerable to egg parasitoids and also to fungi that grow on the egg mass. So we're hoping to make some headway. It's not going to be easy, and we may have to import carefully vetted, studied natural enemies from Asia where they're native. So I'm hoping they're pretty, but they're pretty darn destructive, especially vineyards and some related woody fruit crops. They love tree of heaven, but unfortunately they love some other things too.
They're all over Carroll County, I can tell you that.
Yeah, but they're just getting in. I mean, I've only seen just a few reports from Prince George's, for instance. But they
Yeah.
move rapidly. They're very good hitchhikers. Tracy Lesky up at the Carneysville, West Virginia ARS lab, they simulated in a wind tunnel how fast could you drive your car and then they would finally fall off. And it's above freeway speed, so they're not falling off your car. They love to hitchhike. And if you look at the patterns of colonization in places like New York, they follow the freeways, right? The interstates right out there. You can see the new colonization. So they're going to be in a lot of places pretty quickly. All right. There's my leaf-footed bug shot if you wanted to see some pretty bugs, right?
Oh, yeah, those are nice.
Yeah. I could give a whole nother talk on leaf-footed bugs, but I'm not going to. And
Maybe next year we can get you back again.
right. Well, we'll know more about leaf-footed bugs next year. I can promise you that, Fred.
Mhm.
Okay,
Very
thanks,
good.
everybody. It's been a pleasure, and I hope I answered your questions.
Get a lot of thank yous from the students there, Dr. Weber.
Yeah, I saw that. Thank you. You're welcome. I mean,
Thank you very much for the lecture tonight. We appreciate it, and I can guarantee you for many students, this is something that they've never heard of before, you know, so it was a good exposure to a number of them. And it was certainly very interesting to the rest of us who think we know a little bit about something, you know.
good, all right. Everybody have a good weekend and enjoy your dreams of pretty bugs.
Thanks a lot, and you will be receiving a journal, the Maryland Entomologist, from us.
Excellent.
If I could just get an address where you would like it sent, then
I'll
we can
email that
take
to
care of
you.
that.
Yeah.
All
Yeah.
right. All right, well, appreciate this very much. Anyone else have a question or